No, the excitement contraction coupling is not a second way to increase the strength of contraction. I haven't talked to you about the second way yet, but when I've told you, you will know it. The excitement contraction is just a review of what we're talking about, and that is the way that the skeletal muscle contract is due to the presence of calcium come from the sarcoplasmic reticulum. And calcium binds the troponin, pull tropomyosin out of the way. Okay, and but in this excitement and coupling reaction, what you see the key there is that you need ATP, okay, to causes the uh, myosins to pull out of the actin. Okay. Another way, I think, is probably make more sense if you think of it this way. Um, Contraction is just myosin slide past each other, and so this is actually the slide. Zoom up that area and told you that the once the myosins binds to the actin, it's going to create what we call this right here a power stroke, which is a snap. Okay, a, a power stroke is that once the actin, the myosin grab onto the actin, it's going to cause us a snap like this, a power stroke, and therefore the myosin slide past each other. Another way to think of this is imagine a sailor trying to pull up the anchor. Okay, if you ever go to Pier 39, maybe that's San Francisco, South Street Seaport in New York, right? In doing a fleet week, you might see the sailor pull up the anchor. I guess that's the old day. Now they just machine pull up the anchor. But the sailor when to pull up the anchor, what he does is that he will grab on one arm to the rope, right, and then grab on the other rope, and he's going to pull, right. If you pull, you're going to stick. You have to release this hand, grab onto the other rope, release this hand, grab on the other rope, and then you pull and you release, you grab on, you release, you grab on, and you pull. Because if you just hold on like this together, you're not going to go anymore. Okay, so that is one of the functions of ATP. In order for the myosin to be released from actin with your arms out and grab on the other, you need ATP. So you can see here ATP is the unit of energy. Okay, and we'll see that there are three ways how the body can make ATP. The quickest way is creatine phosphate. Glycolysis is the second way, and cellular respiration is three-way. But ATP is needed for three places. One is to causes the myosins to release from the actin. You can see here, myosin still binds to actin. In the presence of ATP, it's going to cause detachment, right? So that's one functions of ATP. ATP is the energy that is needed to hydrolysis of ATP, will cause it to recock itself, right? Once you cause a power stroke, you want it to set it back out again, right? So it can grab. So to go from here to here, you need ATP. Hydrolysis of ATP causes it to come out, so ready to grab again. Okay, that's the that's the second important functions of ATP. And then finally, ATP is needed to put calcium back into the sarcoplasmic reticulum. Okay, because remember, sarcoplasmic reticulum is an area that has high calcium. This area has high calcium, right? And so if you opens up the calcium channel, calcium will just go out. Now, if you want to put calcium back in, right, this is to go this way is an active process. You need ATP for this. Um, I don't understand. If you have a question, can you raise your hand? ATP is an energy producing or releasing.
Um,
ATP is the energy. The
oh, yeah.
energy is needed for this hydrolysis.
Okay.
Right? Now, you only produce ATP through three processes. Okay, ATP are generated. ATP um, are generated from in the skeletal muscle. What you see is that the creatine phosphate is the quickest way. You know, in order, like when you're running, the first 15 second or so, you get this ATP from this creatine phosphate. I'm going to show you a little bit. And, maybe 30-second, 45-second, you get your energy through glycolysis. Okay? Glycolysis is to break down glucose to pyruvate. Okay, you only generate 2 ATP, but you can do that in the cell cytoplasm, and it doesn't require oxygen, so we call that under anaerobic respiration. And the third way is cellular respiration. And in cellular respiration, this is occur within the mitochondria, but you need oxygen. Okay? You need oxygen. Okay, so I thought I saw a message or something. um, okay. So in the sliding filament model, again, it just basically actins and myosin slide past each other. And doing the contraction, as you can see here, the 8 zone will get smaller, right? Here's the 8 zone, will get smaller. And here's the I band. The I band will get smaller. So remember HI, doing the contraction, two area gets smaller, the H and the I. Um, and so if you continue with this cross bridges cycling with the coupling, I explained to you already how ATP is needed to bind to myosin, and the split of ATP energy is required for that recocking. um, I still only told you one way to increases the strength of contraction is to motor unit recruitment. Okay. I haven't told you the other one yet, so don't worry about that. And I will tell you. Let's continue on with this slide. And this idea now is that when you have a muscle contraction and then you have a relaxation, what is key event that you see in relaxation? The key event is that this enzyme, acetylcholine esterase, decompose or break down acetylcholine. Okay, and without acetylcholine, the stimulus from the sarcolemma contract, so there's no more um, depolymerization down the T-tubule. More importantly, calcium has to move back into the sarcoplasmic reticulum. This is the active process. Requires the ATP. Okay, and then here's the major event that you see in contraction and relaxation that was summarized for you. I don't need to go over. So the important thing, let me talk about how you generate uh, ATP, and then we'll come back to look at uh, the muscle physiology. Okay, there's three ways to generate ATP. One of them is the creatine phosphate mechanism, which is really creatine is a protein that carry a phosphate to this. It's a simple exchange reaction, if you will, so that if you if your ATP is low, right, you can just take the once you hydrolysis ATP will become ADP, by the way, right? This one right here will become ADP, with ATP for adenosine triphosphate, ADP is adenosine diphosphate. So you lose one phosphate. So by hydrolyze that one phosphate, that's the energy it's need to to do the recocking, or if you will. Now, if this ADP is high, you can make more ATP by taking it from the creatine phosphate. So the creatine phosphate will donate that one phosphate to make ATP, and then it loses uh, the phosphate from creatine that become creatine. Okay, and this is only you see a lot in the skeletal muscle, and degradations of the creatine. When we talk about the urinary system, this is broken down to creatinine. Okay, and creatinine is one of the nitrogenous compounds that it has to get rid from our body through the urinary system, get rid of creatinine. So in the future, when we're talking about creatinine, you know that come from the creatine phosphate, which you see a lot in the skeletal muscle. In fact, if in a bodybuilder who who has a lot of muscle turnover, you might see a lot of creatinine in the urine. Um, does taking creatinine help building muscle? I would say that yes and no. In fact, if you want to build muscle, all you need to do is take a lot of protein, okay? And creatinine is just one of those proteins. It's going to get broken down to amino acid and rebuild it, right? And so rather than spend, you know, certain amount of money to say, oh, I wanted to get creatinine in my body. Well, if you ingest creatinine, the final product that your body sees is not creatinine, but amino acid. So that's the same thing as saying, okay, you know what? I'm just going to boil some eggs and eat some albumin, the white part of the egg. That provides sufficient protein for me to need it to build muscle. That works just as fine. So you don't need creatinine, a specialized protein for building muscle. Although creatinine is what you see in the muscle. Um, so take home message, the quickest way to generate ATP in skeletal muscle is the creatine phosphate. Okay. The second way is known as glycolysis, okay, which is this. Now, glycolysis involving 10 steps, and I don't expect you to know these 10 steps. In When I took biochemistry, I have to know these 10 steps. In other words, glucose first converted to glucose 6-phosphate, converted to fructose 6-phosphate, converted to fructose 1,6-diphosphate, which is broken down to glyceraldehyde 3-phosphate, and dihydroacetone phosphate to phosphoenolpyruvate, and then to pyruvate, for example. Okay, so there's 10 steps that is involved to get from glucose to pyruvate. Okay, now pyruvate is a 3-carbon molecule. So one glucose, you will get 2 pyruvate. Right. And here's the interesting thing, and this question I ask my student, and I always the one that know what I'm talking about are usually my A student, and the one that who doesn't get this right, they're not an A student. Okay, so in glycolysis, okay, is a 10-step process. That's all you need to know, which glucose is converted to 2 molecules of pyruvate. Okay. In fact, you know what I'm going to do? I am going to share with you my screen, share screen. All right, so you can just go here. That's the happy birthday song. I want to show you something. So basically, if you Google glycolysis, okay, glycolysis, and you can just say images, and here are, yeah, there's many things. All right, let's hear. So you can see that the 10th step that I remember I told you is that glucose convert to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6-phosphate to dihydroacetone phosphate to glyceraldehyde 3-phosphate to 1,3-biphosphate glycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenolpyruvate, and pyruvate, right? That's the 10th step in glycolysis. In fact, you probably can do here. This is a good one. All right. So you can see 10 steps here. Now, the take-home message is that one molecule of glucose will give you 4 ATP. Okay, so once you get here to glyceraldehyde 3-phosphate, the glyceraldehyde 3-phosphate can continue. This kind of molecule, the other 3 carbon molecule that come up glucose, the dihydroacetone phosphate, cannot continue unless it's isomerized. It becomes glyceraldehyde 3-phosphate. So in other words, this is a 3 carbon molecule. If you go this way, you generate 2 ATP. And you take this, right, and you bring it over here, you get another 2 ATP, right? So in glycolysis, you generate 4 ATPs, but you use 2 to prime it, right? Here, convert glucose to glucose 6-phosphate, you need 1 ATP. Fructose 6-phosphate to fructose 1,6-diphosphate, you generate, you need another ATP. But the net is at only 2 ATP, right? The other important thing is that it doesn't say it here. Oh, here. Okay. Now, in glycolysis, you do make this molecule here is known as NADH. Where does NADH come from? NAD positive. Okay. Now, why do you make NADH? Well, NADH is electron carrier, if you will. It carries the electron to the electron transport chain, which is you find it in the mitochondria. Okay. I know that's a lot, and some of you may not have a lot of biochemistry, but I'm going to make it really short for you. I'm going to stop the sharing, and the take-home message is this. I want you to know is this. Okay, you have glucose, and I'm going to write okay, breaks it down 10 steps, so you don't need to know everything in between. Okay, this to pyruvate. Right? That's not too bad, right? Glucose to pyruvate. Now here's the thing is that I want this to come out is that you generate only 2 ATP as a net, and you generate 2 NADH. Okay, and in order for you to make NADH, you need a molecule called NAD positive. Okay. The difference between NAD positive and NADH, and that is this one right here has a hydrogen ion or electron, if you will. And so NADH you can think of is, by the way, this is 2 NADH, right? So then this I want you to know is electron carrier, right? So NADH brings electron to the electron transport change, which is, I'm going to call this electron transport chain, ETC, which is part of the mitochondria. Mitochondria. All right, so everyone clear so far? This is glycolysis. Right. So the question is, what is glycolysis? Well, you can say glycolysis is the breakdown of glucose to pyruvate. You generate 2 ATP and 2 NADH. What is NADH? Is electron transport carrier. It brings us the electron to the electron transport chain, which is part of the mitochondria. Okay, are there any questions? Okay, there's no question. Let's continue. Now, as you can see, glycolysis is an anaerobic reaction. It does not require any oxygens at all. Right? This is in anaerobic phase, does not require glycolysis. Just occur in the cytoplasm, produce little ATP. In fact, it only produced 2 ATP, and write it down, and 2 NADH. That's key. Now, pyruvate is 3 carbon. Before it goes into the mitochondria, it's loses the carbon dioxide, now becomes 2 carbon called acetyl, which is basically binds to acetyl-CoA. And it's part of this Krebs cycle, or the citric acid cycle, and that is acetyl-CoA binds to a 4 carbon molecule known as oxaloacetate, which then converted to citrate to isocitrate, loses a carbon to alpha ketoglutarate, which then loses another to come succinyl CoA or succinate, fumarate, malate, and then back to oxaloacetate. Okay, you don't need to know this Krebs cycle. I know that because that's my major, biochemistry. And if you don't know that, I will show you what does the Krebs cycle look like. Okay, so as you can see, I have to share with you again. Okay, so you can see here, here's the Krebs cycle. And it's you can see how pyruvate loses the carbon dioxide, become acetyl-CoA. Acetyl-CoA uh, converted with acetyl which is a 4 carbon, to become citrate, to isocitrate, to alpha ketoglutarate, to succinyl CoA, to succinate furanate malate, and come back. So these are the intermediates of the Krebs cycle. But ultimately, what it does is that it breaks down the acetyl CoA into carbon dioxide. Okay, so the byproduct, or the product, the end result, is carbon dioxide. But note, you get this electron carrier NADH, NADH, and NADH. And in the Krebs cycle, you get NADH and a very similar molecule called FADH, which is here. Uh, there's an FADH to come out over here somewhere. Um, and the NADH is the one that is going to generate the most ATP, okay? And that is through the here. I mean, okay, oh, sorry. This is just shows you the NADH, NAA, and FADH, which is similar to NADH. That's the whole purpose of the Krebs cycle. But I'm going to show you now is the we'll call it the electron transport chain. Okay. So here's the electron transport chain. The NADH molecule that you generate, right? And what that does is that it brings the electron to this first component of the electron transport chain. The electron transport chain finds within the mitochondria. So you can say here is that the inner mitochondria and the outer mitochondria membrane matrix. Okay. Now you can see here NADH, give it to the electron, become NAD positive again. And the electron is then cycle, if you will. Well, this is like considered like a water wheel, but in the process, what you do is that you pump hydrogen ion from one component of the mitochondria to the other. And when these hydrogen ions come back in, that's how you generate the ATP. Okay. Here, you can see how the hydrogen ion come back in. That's how you make ATP. Now, that's a lot of detail, right? You can read up on that, but I want the take-home message is this. Okay. Okay. Now, within the mitochondria, right, the electron transport chain essentially makes this is like a water wheel, okay, and the the molecule is known as NADH, who carries the electron to the electron transport chain, right? And when it does, uh, it's sort of like a water wheel where they cycle the electrons through different components of the electron transport chain. But the question is, what is the final electron recipient? Okay, and the final, let's say the final electron acceptor is oxygen. All right, it's oxygen, which is converted to water. So you can see here is that if you don't have uh, oxygen, this thing will stop. This thing will stop. Because once you put the electron into the system, you have to have a way to take it out. And the oxygen will takes that electron and convert that to water. Okay? But if you don't have any oxygen under anaerobic respiration, and this system is blocked. Right. So let me review. In glycolysis, anaerobic respiration, you only generate little ATP, 2 ATP in fact, and 2 NADH. The bulk of ATP come from cellular respiration, which is involving this oxygen called the aerobic phase, which involve the citric acid cycle, which is this, the electron transport chain, which is this. This occurs within the mitochondria, and you produce the most ATP. Okay, and oxygen is needed to keep the system going, because without oxygen, this thing will stop. So in fact, if you if you exercise a lot, right? Let's say if you're running a mile, a couple miles, where your intakes of oxygen is not sufficient, right? So what you're going to do is you're going to be in a condition known as the oxygen depth. And oxygen depth is that you're going to spend the making ATP through mainly glycolysis because there's no oxygen, right? No oxygen, this whole thing stops. Okay. But under anaerobic respiration, your body will convert it to lactic acid, right? And for many years, as people say, why does under anaerobic respiration, pyruvate is converted to lactic acid? Okay. And the answer is that in glycolysis, you generate a lot of NADH. Okay. In order for you to go from glycolysis, in fact, one does not think of it, but you do need this molecule, NAD positive. You need NAD positive, right? Not only do you make this, right, which is we know, but that you need NAD positive for glycolysis to go. Okay? Everyone's get that? You need NAD positive for glycolysis to go. Because normally in glycolysis, you make NADH. And if you have a lot of oxygen, NADH is going to the electron transport chain. It gives off that electron. So then now it can convert back to NAD positive. And this NAD positive can keep the glycolysis going. But if the electron transport chain is stopped because you don't have any oxygen, this will build up, right? And if you build up, then you have no NAD positive. If you don't have any NAD positive, glycolysis will stop. But you don't want that. You want the glycolysis to continue going. Right? So thus, the reason why pyruvate is converted to lactic acid, because it's the way for the body to use up NADH here, convert it to NAD positive. Okay. So we call this a redox potential, because what happened is that pyruvate is reduced and NADH is oxidized. So it's a redox potential. So another way of saying is that pyruvate is converted to lactic acid so that your body can use NADH. Your body uses up NADH to generate NAD positive needed for glycolysis. So that's why. So the take-home message is this. Under anaerobic respiration, your body makes lactic acid. Okay? So you want to understand physiologically, why does it make lactic acid? It makes it so because it's an outlet, a way for it to use the NADH, because you have too much buildup of NADH and you want to convert that to NAD positive, right? So we can see that in glycolysis, you need NAD positive to start going, because I showed you earlier. I'll show you again, glycolysis. Share application screen. All right, so let's see. So not even knowing all the intermediates and stuff like that, but just appreciate, say here, you can see how this molecule here, NAD positive, right? NAD positive is needed to keep this thing going, because if you don't have NAD positive, forget it, this reaction is not going to go, right? But in doing so, you make NAD positive. If you have NAD positive, that can bring that to the electron transport chain. But if the electron transport chain is not working, you're going to have a buildup of NADH. So let's say, again, electron transport chains, and what is the final recipient of the electron transport chain? Yeah. That's stupid. That's you can read it. You can look at the electron transport chain through that. Doesn't matter here. You see how NADH is bring to this NADH is bring to the electron transport chain. It converts to NAD positive. So this is the normal physiology. And what is the final electron transport? The electron acceptor is oxygen. So if you don't have any oxygen, this thing stop. You're going to have a buildup of NADH, and the buildup of NADH will stop glycolysis. Glycolysis can only occur if you have NAD positive. So that's why pyruvate is converted to lactic acid, and I'm going to write it to you here one more time. So let's say pyruvate converted to lactic acid, NADH. And you can see the chemical reaction here. You see that? Here. You can see how pyruvate is converted to lactic acid. You need NADH. NADH is converted to NAD positive, right? You see that? So that's why under anaerobic respiration, pyruvate or pyruvic acid, same thing, converted to lactic acid. All right, so I hope I summarize this for you. The important steps involved in cellular respiration and glycolysis. And the and one of the reasons why you converted to lactic acid is because you use NADH. So in oxygen depth, and oxygen depth is that once you use up a lot of ATP due through glycolysis and conversion of pyruvate to lactic acid, right? Now you got to pay back. Okay? Your depth, now you have to pay back. And what happened is that the lactic acid is found in the skeletal muscle. That lactic acid needs to go back to the liver, where it can be converted to back to glucose as part of this process known as gluconeogenesis. Gluconeogenesis is to generate a glucose, but from lactic acid. A human is not like plants where you can take carbon dioxide and water and sunlight to make into glucose as part of the photosynthesis. We can only make glucose from the intermediate, which is lactic acid. Okay. But another important things that I also want you to note is that lactic acid is an acid. So in other words, pH is going to be low. Okay? So one of the reasons why under muscle fatigue that you don't have an uncontrollable reaction, and that is because of the pH imbalance. Right? There might be too much ion so that the interfere with the flow of potassium, for example, which can repolarize the membrane. That's why you get uncontrollable reaction. Um, Later on, I will tell you too that one of the ways to get the blood to flow in the venous circulation is that you need to have skeletal muscle contraction. Okay, and what I mean by that is imagine that um, here's a muscle, here's a skeletal muscle, and here's another skeletal muscle. In between the skeletal muscle is a blood vessel, let's say vein, okay, vein like that, and blood will flow in the vein, right? So I'm going to draw this as blood. Now, let's say that it's going to contract some more. If it contracts some more, contraction you can say is that you can shorten the sarcomere. So shortening the sarcomere is going to look like that. Shortening the sarcomere is going to look like that. And now your blood vessel almost like it's occluded. Okay. Stop. And then muscle relaxation, going to look like that. Now the blood vessel can flow. You see? So the presence of the skeletal muscle contraction help blood flow. Now this is important because if you're running, say 5 miles, right, and you're going to build up lactic acid in your skeletal muscle, an athlete will know that after the run, you don't stop. What you do is you do a warm down. Okay, a warm down is a way to still causes the muscle to contract to help brings the lactic acid in the skeletal muscle back to the liver. Within the liver then can convert it back to glucose. Because if you don't, the lactic acid will accumulate in the tissue and then you can have muscle fatigue. But more importantly, you probably feel the pain the next day. And so the warm down, it's a good process to still causes the blood flow. Okay. Another important thing to note also is that um, doing skeletal muscle contraction. This is during. So I'm going to. Right. And this is actually there's a couple called the Cori cycle, which is they won a Nobel Prize for this. And I'm going to show you. It's known as the Cori cycle. Okay. And what's the Cori cycle entail is this. Okay. So let's say here's the skeletal muscle. And skeletal muscle contraction will build up a lot of lactic acid under anaerobic condition. Okay. Now, through the cardiovascular system, lactic acid is getting to the blood. The blood is going to go to the liver. Okay. And lactic acid like that. That's it. It's going to convert it to glucose. Okay, glucose. Now, what Cori cycle does is that now a lot of cells once they take in glucose, that glucose is changes automatically to glucose 6 phosphate. So it just add a phosphate to that, and the glucose get trapped. It cannot go out. So in other words, most of the cells when they take glucose, they don't export it. But what the Cori cycle just they discovered that um, uh, liver cells, right, hepatocyte, have the capability to release glucose. Okay, so they not only import, but they export glucose out. Okay, and glucose then get back needed for the skeletal muscle contract as well as glycolysis. And so you can see this is it's known as a cycle, is because the lactic acid is put in the blood. The blood goes to the liver converts the glucose, the liver releases the glucose back into the blood, and it goes to the skeletal muscle. That's a beautiful um, Cori cycle, as you will. Okay, how glucose can leave the liver. Uh, in fact, we know this, in general, if you have too much glucose, that glucose can store it in terms of glycogen. And if you glucose, you can break glycogen, and then glucose can exit the hepatocyte and dump into the blood. Um, so that glucose then can not only import, but also export it out of the cell in the liver cells. And the glucose is entered the blood, within the glucose can bring it to the skeletal muscle is needed to keep the glycolysis going. That's the Cori cycle. All right, I'm going to stop here. Are there anyone have questions? We have one more hour, so we should finish up with the skeletal muscle, the uh, uh, muscular system for your exam next week. Okay. All right, so I'm going to stop here.